here. So in this experiment, he grabbed these flower, that are purple color, these purple flowers, and he cut the stamens. Notice here that they don't have the stamens. So they're not able to produce their own pollen, okay? So self-pollination is not an option, okay? So they only have the carpel right there, the egg producing cells right there. So he grabbed the pollen, you can see here from the stamen, from another plant, in this case, the white flower. So he grabbed the pollen from the stamen and put them into the carpel of this purple flower. So he grabbed the pollen from the white flower and he put them into the carpel of the purple flower. Then the pollinated carpel gave rise to the mature pod, and then he planted these seeds from the pod, and then gave rise to the F1 generation. So that will be mixed hybrid. Okay, parental generation, F1 hybrid. He mixed these two. Then he saw them that they were all purple. The white, no longer there. Okay. So we're going to go back in a minute. So he then tracked only these characters that occur in two distinct alternative forms. So he used these flowers that only give rise to either purple or white. Okay. And he used these varieties that were true breeding plants. In other words, they produce offspring of the same variety when they self-pollinate. In other words, these are true breeding. This parental generation, this purple flower were true breeding, and these white flowers were true breeding. True breeding basically means that they are homozygous. Okay. They are homozygous for the purple color, they are homozygous for the white color. Therefore, these flowers can only give rise or can pass on this trait, purple colors. Their offspring can only be purple, nothing else. And these white flowers, they can only pass the white color trait to their offspring. So, if you self-pollinate these white flowers, they will not be able to give rise to purple flowers. If you self-pollinate these flowers, purple flowers, they will not be able to give rise of white color. They can only give rise to purple color, they can only give rise to white color. They are true breeding, they are homozygous. Okay?
Like a true breed dog.
Yes, that was the example right there, right? Like true breed dog. If you cross like lap and lap and you get a chihuahua, they're not.
It's a mutt. That's like F1, right? That would be F1.
Yeah, no, this is not like true breed. Yeah, in that case that would be like if you mix them, right? In this case, then that one would be a heterozygous from these two, which are not pure breed. Okay, so they will like mix, they would like hybrid. Okay, but here in Mendel's experiment, he used this hybridization from true breeding varieties. Just keep that in mind. They're pure breed. Okay. Yes.
So like capital P, capital P.
Yes, that would like homozygous dominant. That would be for example, the purple. And then just recessive, homozygous recessive for the white color. Okay. But here, you have here the example. Okay, that is the parental generation, homozygous. That is the true breeding. Okay. And then, by allowing for this, then you have this process of hybridization. Whenever he mixes them, okay. Whenever he mixes them, then those hybrid generations, those are called the F1 generation. The true breeding parents, those are the P generation, homozygous. Homozygous dominant, homozygous recessive. Then once he mixed them, the true breeding, then the hybrid generation, that is the F1 generation. And then whenever he allowed for this F1 generation to self-pollinate or cross-pollinate, then that gave rise to the F2 generation. Okay. So here you have the Mendel's experiment. So you have the parental generation, these are true breeding parents, purple flowers, white flowers. Purple flowers, homozygous dominant, capital P, capital P. It cannot give rise to white flowers. And the white flowers, homozygous recessive, little p, little p. Okay? It cannot give rise to the purple color. So those are homozygous dominant, homozygous recessive, true breeding. Then he mixed them, right? He put the pollen from the white into the carpel of the purple, and you have the hybrids, that is the F1 generation. And here you saw that all plants were purple colors. Then whenever you allow this F1 generation to self or cross-pollinate, then you have the F2 generation. And on that one, he saw a 3-to-1 ratio of purple color to white color. Okay? And that's what we call the terms, dominant and recessive. Where dominant is the purple, homozygous dominant, right there. And here you will have some mix of homozygous dominant and has some heterozygous, and that's why you see a bigger ratio, 2 to 1. And here you will have the homozygous recessive, little p, little p. Okay, that's why he called that recessive, because here you are not seeing the white color, but it doesn't mean that it's being destroyed or eliminated, but rather that it is being masked or like it is not being able to be expressed. Right? That's the reason of the dominant allele. It's not allowing for the recessive allele to be expressed.
So some of the purple flowers are big P, and some of the purple flowers are big P, little p.
Heterozygous, yes. So the reason is that only the purple flower factor was affecting the flower color in the F1 generation that we have here, right? That is being the dominant allele, not allowing the little p to be expressed. And Mendel called this flower color a dominant trait, while the white color, it was a recessive trait, only expressed whenever it's homozygous recessive. That's when you have the expression of this recessive trait, okay? The factor for white color was not diluted or destroyed, nor eliminated. Why? It reappeared in the F2 generation. That is one of the main key points for this recessive trait. It reappears, and only is expressed whenever it's homozygous recessive. Okay, so some key terminology, right? Dominant trait and recessive trait, right? The dominant trait, the one that is expressed as long as it has one dominant allele. It can be homozygous, or it can be heterozygous. Well, as long as it has one, then it's going to be expressed, that dominant trait. Recessive trait, it needs to have both recessive alleles in order to be expressed. Okay. So he observed the same pattern of inheritance of another 6 other pre-plant characters, each represented by these 2 traits. Okay. So he called these heritable factors, as we mentioned in the beginning, but now we call them genes. Okay. What are these other characters or traits? Then you have here the ones that we showed, the flower color, purple and white. He also work with the seed color, yellow and green, and the seed shape, one and wrinkle, and also like pod shape, pod color, flower position, and stem length. Many other people like characters. So he developed this hypothesis to explain the 3-to-1 relation pattern that he observed in this F2 generation. He coined these 4 concepts to make up this model, and these concepts can be related to what we know now about genes and chromosomes. So the first one, alternative versions of genes account for these variations in inherited characters. That's what we mentioned before. Alleles are alternative versions of the genes. So point number 1 of these 4 concepts. Right? Alternative versions of the genes accounts for this variation on those characters that are inherited from one generation to the other. So with the gene for flower color in pea plant exists in 2 versions, one for purple flower and the other one for white flower. That was the example here by the 2 students. Right? Here is the in the region of the chromosome that it codes these genes for the flower color character, and each of these alleles it codes for a variant of this character, a trait, either purple or white. That's the allele, alternative versions of the gene. That's what we have there. And each gene resides in a specific locus and specific chromosome. That was the point that I was trying to make. On this chromosome, this region right here, it codes for flower color. Flower color here, flower color there. Okay. But this chromosome has here this allele where it expresses for the purple color. This other region right here, allele for the white color. Okay. So alleles, alternative versions of a gene. So that's point number 1, alleles. That we have there, for example, here we have these pair of homologous chromosomes, right? In this chromosome that let's say comes with that, you have here, you have in this chromosome the location that codes for flower color, right? That is the gene right here, flower color. But here on this chromosome, you have the allele that codes for purple flower, whereas in this other chromosome, you have the allele that codes for white flowers, okay? Notice that. Here is the gene location that codes for flower color, character, okay? In these homologous chromosomes, okay? But here you have the allele trait that codes for the flower color. That is the variant in the character, okay? The trait, in this case, purple color. And in here, you have the allele that codes for the white color, trait, okay? Does this now make more sense? So gene, right here, the region of this gene is for flower color, character, right, big umbrella. And then there is character variance, so that is the trait, white, purple, right? So that's the allele. The allele codes for the trait, white color. The allele codes for the trait, purple color. But both are flower color character. Right? There is a gene right there. The allele is a specific color. Okay? Does that make more sense? Right, okay. Point number 2. For each of these characters, an organism inherits 2 alleles, one from each parent, right? So, Mendel made this deduction without knowing about the role of these chromosomes. These 2 alleles, particular locus, may be identical, in case they're like homozygous, as in the case for the true breeding plants in P generation, or they can be, for example, these heterozygous. They can differ, they can be mixed, okay? They can be like one dominant, one recessive. So, these characters, you can have like variations, okay? But you inherit one from each parent, okay? You inherit one allele, one from the mom, one from your dad, okay? Let's say, for example, here in this example, one allele is inherited from mom, one allele is inherited from dad. So they could be either mixed or they could be the same, okay? If they are mixed, then the one that is dominant, it will be expressed, okay? If they are the same, then both of them will be expressed, homozygous, okay? So, first concept, alleles, alternative version of the gene. It codes for the differences in the character variants, okay? Second concept, for each of these characters, you receive 2 alleles, one from each parent, one from dad, one from mom, okay? So, concept number 1, alleles, alternative version of the gene. Concept number 2, you receive one from mom, you receive one from dad. Okay. Third, if the alleles at the locus differ, then one, the dominant allele, will determine the appearance, the phenotype. That's the one that is going to be expressed. The other one, the recessive allele, will not be expressed. It will not be noticeable until it is like homozygous. So, in the flower color example, the F1 plants of the purple flowers, they show this because the allele is dominant, the purple color. Okay? So, that is the key point of the third concept. So, first concept, what is it?
Alleles.
Alleles, alternative version of the gene. This goes for the character variants, the traits, right? Concept number 2.
Character.
One from mom, one from dad. And you receive 2, one from mom, one from dad. Concept number 3. If they are different, the dominant will be expressed, right? The example right here. If one is capital A, one is lowercase a, the big A will be expressed, the other one will not be expressed, okay? If the little a and the little a are there, that's the genotype, then you will have a recessive trait, okay? If you have big A, homozygous dominant, you have big A, little a, heterozygous, but you will have a dominant phenotype, okay? So, third concept will be like the dominance, the expression of those alleles, which allele will be expressed. In this case, the dominant. The recessive one will not be expressed unless it is homozygous recessive. Okay? And then fourth concept, now it's known as the law of segregation. So that means that you receive 2 alleles, one from mom, one from dad, but during the gamete formation, they will separate, they will segregate, and you will only give one to your future offsprings. Why? Because the other one will come from the other parent, right? So if you are the mom, you will give one, the dad will give the other one. If you are the dad, you will give one, the mom will give the other one. So law of segregation, you only give one of those alleles. If you have 2 alleles, one from mom, one from dad, one is dominant, the other is recessive, you only give one. Law of segregation, you could either give the dominant or the recessive one, right, if you are heterozygous. Okay? So this.
Sorry, I have a question with that.
Yeah.
That applies to blood type, right?
What was that?
That applies to blood type, right?
Yes, blood type, but the blood type is different expression of dominance. This one is complete dominance. You will see an example over there. That will be more like
Codominance?
codominance.
Okay.
Yes. So it will be. It's not as simple as this, either dominant or recessive. Yes. So this segregation of alleles corresponds to the distribution of these homologous chromosomes to different gametes in the meiosis. So that basically means that how you receive those alleles during meiosis, formation of those gametes, then you will pass those alleles. Right? So you don't determine which alleles you will pass. Okay. All right, so here are the 4 concepts summarized. 1, alleles, alternate version of the genes, that basically account for this character variation. Point number 2, that from each of these characters, the organism inherits 2 alleles, one from each parent, one from mom, one from dad. And 3, dominance, so if they are different, then the one that is going to be expressed, that is the dominant allele, not the recessive one. And fourth one is the last segregation that you want to give one to your future generation. Those that will segregate and you're going to pass one. Okay? So make sure you know these four concepts, right? So let's over here. Table number one over here. First concept, what is it? Gaspar? First concept?
Alternative versions of genes account for
Yes. Alleles, right? Alternative versions of a gene that accounts for those character variants, those traits, okay? Okay, number two, concept number two.
Each character organism inherits two allele, one from each parent.
One from mom, one from dad, okay? Over that, number three. Yes, expression, right? Dominant. Which one will be expressed? Number four?
The law of segregation.
Which means?
States that all the two
Alleles? You want to give one, okay? You want to give one to your child, okay? And you guys always respond, so you don't have to. Alright, so here you have, for the Gosling baby, the picture that I showed you at the beginning, right? What are they going to inherit, these babies, from their parents, right? So here you have the purulocus, to make them beautiful.
What
babies,
Gosling?
right? So here the royal
Oh, yeah,
sperm,
right.
in this case, the royal sperm doesn't have the purulocus, okay, to make the babies beautiful. But here for Eva's egg, it does have the gene purulocus right there. Okay? So whenever we fertilize, then the baby will be beautiful. But all babies are beautiful, so don't follow this. That is true. That is true. Okay. But yes, the point over here is to show you how you got here the gene, right? Goes for the character, and here variant, trait, beautiful, not beautiful, right? And that's alleles. And second concept, it is one from dad, one from mom, right? So he receives one from mom, one from dad. She receives one from dad, one from mom, right? That's second concept. Third concept, expression, dominance. So he has this allele that is dominant, so that will be expressed. So he is beautiful. She is beautiful too. She has the allele right there, right? So dominance, expression. And then fourth concept, segregation, right? It will only pass one. Meiosis, reduction, right? So if one dies, go from diploid to haploid, only gives one. Okay? So in this case, this sperm was not the lucky sperm. It's going to be an ugly baby. In this case, segregation. This egg cell did receive the beautiful allele. The allele that we were beautiful.
But it received it because it was a dominant.
That's right, over here. Didn't receive because it was dominant. In this case, it was dominant here, but this one didn't receive it, right? That is the law of independent assortment. We will see in a minute.
It had a 50% chance, right?
In this case, for that, yeah. But, given all the accounts of all the other, like, chromosomes and this, right? Yeah. It will be different. But yes, in this case, right there, either here or here, 50%, right? That would get it right there. And in this case, it was not the lucky one, but here it was the lucky egg, right there. So, fertilization, boom, beautiful baby. Okay, law of segregation. Right. So here is basically showing you the four concepts for Mendel's idea of genetics, law of genetics. Okay. All right. So here you have the explanation of this 3-to-1 ratio that he observed in the after generation. So he used a Punnett square that basically to be able to allow for this possible combination of the egg and the sperm to see the possible genotype using this Punnett square. Okay. So using this genetic class to predict the genetic makeup of the offspring, okay? By knowing the genotype of the sperm and the genotype of the egg cells. Now you create this Punnett square, and then you determine the genotype for these particular offspring. So that is the Punnett square. Capital letter represents dominant, and lowercase represents recessive allele. So that's what you have there, the Punnett square. That is this diagram of predicting the results of this genetic cross between these individuals of non-genetic makeup. That is, you know the sperm genotype, you know the egg cell genotype. By knowing that, the parents, then you know the genetic makeup of the offspring. Just one sperm. And that we have right there. So in this case, you have here the P generation through breeding, and that means that it's homozygous dominant. P, P, big P, big P. And the white flowers through breeding, that means homozygous recessive. Little P, right there. So Laur's segregation, you can only give one. So therefore here, you can only give one. So regardless, it will be big P. And here, the same thing. Regardless, it will be little P. Okay. That's why they are through breeding. They cannot give anything else than that. So once you mix these two, you have the hybrid generation, F1 generation, so that will be heterozygous, that will be mixed. But because they have the big P right there, dominant allele, they will be purple all the time, 100%. Okay. But then whenever you allow for this F1 generation to self-pollinate or cross-pollinate, then you have the F2 generation. So by knowing this, the genotype of the parents, here the sperm cell, and the egg cell, both will be heterozygous. Then you can determine the ratio, the genotype of the offspring right here. And from that you can determine the ratio. It will be 3-to-1 ratio. 3 purple, 1 white. Okay. This one will be homozygous dominant, this one will be heterozygous, and this will be homozygous recessive. 3-to-1 ratio. So make sure you know that one. Phenotype, those are the physical traits of the individual, the appearance, right? Genotype, that is the genetic makeup, the description of those alleles for those particular genes in the individual. So for example, right there, this is the gene right there. Okay. Well, whenever we show you here the example, that is the gene right here, both of them right there. But here you have one allele, here you have one allele. Both of them make up the gene. In this case, you have this gene that is heterozygous. Okay. And then here something over here. So you have them right there, and you could be like homozygous or heterozygous. Either homozygous dominant, if they are like capital P, homozygous recessive, they are like lowercase, heterozygous if they are mixed. Do not call them, whenever I ask you this, and ask you for example, or like an answer on the question about this, do not call them heterozygous dominant. I have seen that so many times. There is no such thing as heterozygous dominant. Okay. So it will be like homozygous dominant, homozygous recessive, or heterozygous if they are mixed, big and small. Okay. And then here, know this one too. That the recessive, that is the weaker one, that is the phenotype of the allele that is seen only whenever it's homozygous. So another one, whenever it's heterozygous, the dominant allele will sequester, will not allow for that recessive allele to be expressed. Okay? And then the dominant, that is total. That is the phenotype that is seen when homozygous or heterozygous. As long as there is one allele, dominant allele, then it will be sufficient enough to express that phenotype. So that's what you have right there, a 2-to-1 ratio. You are seeing 3 purple and 1 white flower, 2-to-1 ratio. So if I ask you for the phenotype, that will be the answer, 2-to-1. But if I ask you for the genotype ratio, then you will give me this. 1 is homozygous dominant, 2 are heterozygous, and 1 are homozygous recessive. So that would be a 1-to-2-to-1 ratio. So whenever I ask you, give me the phenotype ratio, boom, 2-to-1. Whenever I ask you, give me the genotype ratio, boom, right here, 1-to-2-to-1 ratio. Question with them? So this question may vary, depends on the Punnett square, okay? On that one, it was really easy to see because it was like heterozygous. Heterozygous. On the mom and dad right here, sperm and egg, right? Heterozygous. Really easy to see. 2-to-1 ratio, 1-to-2-to-1 ratio, genetic makeup, right? But what about if it is heterozygous, homozygous dominant? What would be the genetic makeup, genetic ratio? Right? What would be the phenotype ratio? Okay. What about if it is heterozygous, homozygous recessive? What is the phenotype ratio? What is the genotype ratio? Okay. What about if it is homozygous dominant, homozygous recessive? Phenotype ratio, genotype ratio. Make sure you know all of that. So make sure you're able to do the Punnett square. So that was the monohybrid cross, right? Dealing with one gene, 2 alleles, okay? That is a monohybrid cross. A test cross is different, okay? A test cross is going in reverse. Monohybrid cross is, you know the parents, you know the genetic makeup of the parents, you are going to determine the composition of the offsprings, genetic makeup and phenotype ratio, right? The monohybrid cross, you begin with the parents and you determine the offsprings. Here the test cross is using the offsprings to determine the genotype of the parents. Okay, so it's going backwards. So how can we tell the genotype of any individual with the dominant phenotype? Okay, so for example here, if you have here the purple flower, right, the parent one, here you have the white flower, you know that this one is homozygous recessive, little p. But what about this one? How do you know if this purple flower it is capital P, or capital P, lowercase p, right? Homozygous dominant through breeding, or just heterozygous but showing the dominant phenotype, right? Just by looking at the purple color, you can see that, right? You don't know if it is true breeding, homozygous, or if it is heterozygous.
If you rebreed it with homozygous recessive.
So that's what you're doing right here. That's this one. That is a test cross, right? So here you can see this, that you use this one, this little p, and then mix it right there with this purple flower. If it is big P, homozygous dominant, then all the offsprings will be purple, right? If it is heterozygous, big P, little p, then it will be half and half, the offsprings, okay? So that's what you do that. You do this determination of the offsprings to determine what was the genetic makeup of the parent, okay? So that is a test cross. So notice that to the monohybrid cross and the test cross. Why would you use them? Okay. Okay, so that is a monohybrid cross dealing with one gene, two others. But also you can have a dihybrid cross, so that means that you have two characters. So that means there are two genes. And if you have two genes, then how many alleles do you have? One gene, two alleles, two genes,
Four.
four alleles, right? That is the dihybrid cross. And that's what you have right here. Notice here that this one, let's say that this is the true breeding, where you have here the yellow and the yellow dominant, so that is the yellow color dominant phenotype. And you have here the R and R, big R, that is for this seed shape, round, okay? And whereas this one, it is both recessive, lowercase y for both of them homozygous, so that is green, the recessive color. And lowercase r, that is the recessive for the seed shape, wrinkle, okay? So notice there, you have these four alleles, big Y, big R, little r. Four alleles, two genes, the seed color, seed shape, and that's what you do with this dihybrid cross. Now, this is where it comes the law of independent assortment. Somebody mentioned, like, did he pass this on because he was the capital allele or not, right? This is where it comes the law of independent assortment. In this case, if it were to be dependent assortment, that is, that these are going to be passed on, these alleles, because these are dominant, so therefore these will go with all the dominant alleles, they will go together, then that will be the law of dependent assortment. In other words, it depends on the dominance. If they are dominant alleles, they will come together, okay? That's basically what they're showing you right here, okay? These capital Y's, they will come with these capital R's, right? It depends on the dominance. Both are capital, both are dominant, they will come together, right? Same thing here for the recessive ones, they will come here like that. And heterozygous, they will just like mix. That is the law of dependent assortment. But if that were to be the case, whenever you do this dihybrid cross, then you will expect the same thing, a 3-to-1 ratio. Having these both dominant phenotypes, and here having a both of the recessive phenotype, 3-to-1 ratio. Right? However, whenever he did this dihybrid cross, what he saw, it was this, a 9-to-3 to 3-to-1 ratio. Disproving this concept of law of dependent assortment. So it's actually now the law of independent assortment. So in other words, that these capital alleles, they will segregate with either or, capital or recessive alleles for other genes. Okay, that is the law of independent assortment. So they will not like discriminate on the other alleles. If it is a capital allele, it will not discriminate on the other alleles whether they are dominant or recessive, they will come together regardless, and they will be segregated. Okay. So whenever he did this hybrid cross of these, in this case for these two genes, the seed color and the seed shape, he saw this at the end, a 9 to 3 to 3:1 ratio. So 9 double dominant, here 3 single dominant, in this case for the round shape, the color is recessive. 3 of the other single dominant, the yellow color dominant, the wrinkle shape, that is the recessive one, and here 1 out of 16 double dominant, seed shape and color. Okay? That's what we saw there.
Is that for the law of what?
Independent assortment.
Can you explain this one more? I kind of got this.
Okay, come over here.
No.
Yes, come on. Come. Lead you two and you and you. Yes. Come on. Come. Move really quick. Extra credit points. 1 point.
1 point.
Okay. So here we have two alleles for one gene, okay? Let's say that you are yellow and you are green, okay? Yellow, green. Which one is dominant?
Yellow.
Yellow. She is dominant, okay? And you are?
Green.
And yeah, and
Oh.
recessive,
Oh.
recessive, right? Dominant, recessive, yellow, green. You guys come over here, okay? So now they're coding for seed shape. Who wants to be wrinkle? Okay. His wrinkle, right? So that is what? Recessive or dominant? Recessive. Wrinkle is recessive. You are, wow, okay? You are dominant. I was like, oh man, I'm in trouble. Yes, you are round, you are dominant. Right? Wrinkle, recessive. You?
Green and recessive.
Yellow and dominant.
Okay, good, perfect. Okay, the law of what I taught, dependent assortment, basically means that, oh, you know what? I'm dominant, right? Over here. Right? I'm going to segregate, to form the sperm or the egg cell, only with dominant. Come over here. I don't want recessive. I don't want other recessive traits. Go over there, right? Choose one, right? We don't want it. Only dominant, right? So she will be, oh, I'm not wanted, nobody wants me, I'm recessive, I will go with the recessive one as well, right? So that would be like one sperm over here, that would be another sperm over here, right? Yeah, I don't want the recessive ones, right? That is a dependent, right? You can see there the discrimination, right, against the recessive ones. Now what is proven because what took place, what in reality happened, as you can see here, this dominant allele come over here, right? She's like, okay, let's make one sperm. Let's segregate as well the other alleles, right? Here you have recessive, here you have dominant. It can actually go with either one. It can actually go here with the recessive. It doesn't matter, okay? Or it can actually go with her, right there. It's either way. It's independent. It's independent. That means I can go either with the dominant or with the recessive. It is not dependent on like she's being dominant, but it will only go with the dominant one. It can go with either one. Okay? Law if independent assortment. Okay? Does that help?
Yeah.
Okay. 1 point.
I have a question.
Yes.
So he originally thought it was dependent assortment until he started interbreeding?
No, not that he thought, but it was an idea that it was prevalent on that time.
Okay.
But with his experiments, it was disproven.
Okay, so there is actually no dependent assortment.
There is, what I will mention in a minute, is not dependent assortment, but it is the linked genes. So that means for the linked genes, their alleles really close together, that they will behave as one. And that will be the closest I think that you have as dependent assortment. There is no such thing as dependent, but they're like linked genes, they will behave as being dependent.
Okay.
Because they depend on one another, but in reality is that they are really close to one another. They cannot be separated.
Okay.
Okay. So that will be on the exam, but I will mention that more also on the next class. The dependent assortment. So that is the law of independent assortment. So this one speaks about genes who are on different non-homologous chromosomes or those who are far apart on the same chromosome, allowing enough distance to be separated for the crossing over to take place, the tetrads, right, the chiasmata, right, crossing over. Genes who are near each other on the same chromosome, then they are going to be inherent together. They will behave as that, as the dependent assortment. Okay, they will be the linked genes. Okay, so extending this Mendelian genetics, so this inheritance of characters by single genes may deviate from simple Mendelian genetics. This is really simple, right? We simplified, two alleles, one gene, right? But it can actually be more complicated than that. For example, whenever alleles are not completely dominant or recessive, here we just or dominant recessive, but it can actually be more complicated than that. When a gene has more than two alleles, which here we just see like big, it can actually be more. And when a gene produces like multiple phenotypes, not just like, for example, one flower color, right? So it can be more complicated than that. Okay? For the degrees of dominance, here we have the complete dominance. That's the one that we mentioned, like either dominant or recessive, right? The phenotype of the heterozygote and dominant homozygote are identical. So in other words, the homozygous dominant, heterozygous, the phenotype is the same because it has at least one dominant allele. That is complete dominance. As long as you have one complete dominant, then you have the phenotype. Incomplete dominance, that's the phenotype of the F1 hybrid that is somewhere in between the phenotypes of the two parental varieties. So in other words, it is a new phenotype and it's in between, okay? In between those parents, between those parental phenotypes. It's a new phenotype and it's in between.
Like pink?
Like pink for like red and
red and
white.
white, yeah.
Okay, so it's a new phenotype that is in between. So keep point about that, the incomplete, that it is a new phenotype that is from both of the phenotypes of the parents. Okay. The codominance, that is whenever it's neither dominant nor recessive, but rather both are going to be expressed. So those two dominant alleles, they will affect the phenotype in separate distinguishable ways. So both will be co-expressed, hence codominance. Here you have an example of this incomplete dominance, such as here this flower color in the snapdragons. You have the red, homozygous, capital R, and here you have white, the homozygous lowercase r. Okay. Whenever you have heterozygous, then you have a new phenotype that is in between of the parental traits. Okay. In this case, pink. How do you know that this is not blending hypothesis? Remember the blending hypothesis at the beginning?
Mm-hmm.
What was the blending hypothesis?
Where you're both just like the mom and dad kind of like.
You just come together and they get mixed, right? They are just like mixed in their DNA. How do you know that this is not blending hypothesis?
Because it's when the dominant one has to like, it's not really like that dominant, so it kind of just like, oops.
Yeah, kind of like that, right? So one way to know that this is not blending hypothesis, because the blending hypothesis basically says, yeah, you have the genetic
It's just
makeup.
not fully expressed.
You have to
Right.
cross the pink with other
Yeah,
pink.
yeah, yes, and then see
It's the coding.
that you are able to see the phenotype of the parents, right? If it were just to be blending hypothesis, if you mix pink with more pink, then what do you get? Like mixture of pink, right? Hibiscus or whatever, no? Different colors. But whenever you mix these heterozygous with one another, then you are able to see that you restore the phenotypes of the parents. You will see some red, you will see some white. So it's not blending hypothesis, right? So another way to disprove the blending hypothesis.
I have a question.
Yes.
So, sorry. So when people have like different colored eyes, like a blue one and a brown one, would that mean it was like the
Different colors.
Like which one? Like incomplete?
some? It's because some dogs or like some people have like different colored eyes. So one is blue and the other one is brown. Would that be like complete dominance or
No,
that's a
no.
whole different thing?
Yeah, that would be like
I-
something like that would be either, for example, like mutation that we have in there, or something that went wrong in the full expression of those eyes. Let me look more into it. But yes, some of these genes. I mean, this is really simplistic, right? Just one gene to alleles, right? But in fact, like eye colors, eye colors, hair colors, like the height of one person. Oh, God. It involves like maybe like 700 alleles. So it's
Like
not.
polygenic.
Hmm?
The same polygenic.
Yeah, polygenics. And you have like polymorphism as well. And yes, so it's not as simplified as this. Right? This is just to understand this concept of Mendelian genetics. Imagine that, right? Imagine if it were simple like that, right? For like human height. Oh yes, you are here, you are 6 feet. If you are here, you are 5 feet. If you are here, maybe you are like 5.5, right? And all humans will be just like this size, this height, right? Either like 5 or 5.5 or like 6 feet. It is not. It is not. There's like so many. Yes, you have some.
I thought
No, no, and I don't know if you have seen it, what was the Chinese player, basketball player, Xiao Ming? Yao Ming, right?
In the rock.
Xiao Ming. So Yao Ming, right? Imagine that the parents, you will see them like right here. You know, so it's not, right? It's many different, it's many different patterns that there's going on. Okay, the other one codominance, so neither allele is dominant to the other, so you can have, like, for example, here the heterozygous will show both traits at once. In this case, the code color in cattle. Here you have the R1, R1, here shown here in red, and here the R2, R2, shown here in white. And then you have here the heterozygous, and you can see that it's mixed, patchy, okay, red and white. So codominance is both. Think about the blood types that we did in the lab, right? Codominance, A and B, A, B group, okay. And then we go to the pedigree analysis. This pedigree is a family tree that describes this interrelationship of parents and children across these generations. And it will show you this inherited parents, how these particular traits are being passed on from one generation to the other. Okay? With those, you can determine the mode of inheritance, whether it's going to be a dominant trait, whether it's going to be a recessive trait, whether it's going to be a sex-linked trait, or autosomal or mitochondrial terminal effect. So you can determine this probability for affected offsprings for a given cross. Make sure you know these symbols: square, white unaffected, square, black, affected male. Here you have female, unaffected, and affected female, unknown sex, and death. Okay? But for this one, question number one, individual 2 and 5, in the pedigree shown to the right is what? Right here. 2, 5. A, B, C, or D?
What? Individual.
In this individual right here,
B?
a male who does not exhibit the trait.
So B. No.
But it is a male who doesn't exhibit the
What about A male who does not exhibit the trait? Is that true or not?
trait. Yeah.
Yeah, that is true, right? This one is a male affected. A male showing the trait. Okay, so this one is unaffected.
So it's A.
B, A is true. B, is he a father?
Yeah.
Yeah, you say that he's a widow, but yes, he's a father, right? Yes, still human.
Also, all of the above.
C, he has 3 affected siblings? Yeah, 1, 2, 3. Yeah, so that will be all of the above. Right? So make sure you know questions like that.
Like
will
how to
come
read
inside.
it?
Hmm?
Like how to read it?
Yes. Questions like this in the exam, make sure you know how to read it. Right, here's another example. So here you have the affected male, affected female in purple. So here you have one. This trait is for the widow's peak, right here, widow's peak. That is a dominant or recessive trait. Okay, so you can see here that whenever you have here these heterozygous, you have affected children, and then here you have these 2 affected parents, and then whenever they show an unaffected child, then you know that this is a dominant trait. Okay, affected parents, affected siblings, but there is one unaffected child, and that is a dominant trait. That's what you have right here. Okay, affected parents, affected siblings, but one unaffected child, that is a dominant trait. Why is that? Notice here, all of them are affected, right? So, what is the percentage of affected in this case? 75% affected, right? Unaffected, 25%. So that means the majority.
That's like the 3 to 1 affected population.
Yeah, in this case, right? But it's not like in terms of like the ratio 2 to 1, because in this case you only have like 2 child, but so in that case it will be like 50-50, right? But yes, whenever you have this case, in this case you have the unaffected, and then you have the other children affected. Parents are affected, so the majority are affected. So whenever the majority are affected, dominant trait, because it only takes one allele to be this, right? To be expressed and to have the trait. So dominant trait. On this case, you know, we have here the attached earlobe. That's why there you have the free earlobe. What's that?
I laughed because we both grabbed
You
our
have it? Oh,
ears.
you got it.
Yeah,
You
no,
have
we
it.
grabbed our ears as soon as you
You're like
said
testing?
that.
Okay.
Yeah.
So that's what you have right there, and then here you have 2 unaffected parents, and then here you have 1 affected child. So that means that it's going to be a recessive trait, okay? So in this case, if you have unaffected parents, unaffected children, but one affected child, and that means that it's a recessive trait, okay? So in this case, what is the percentage of the unaffected? In this case, you have here that there are 75%, for the child, unaffected. And 25% affected, okay? Having the trait, okay? So the minority, in this case, are showing the trait, the ones that are affected. So in this case, if the minority are the ones showing the trait, that is a recessive trait. Okay. The other ones are protected because it will take just like one dominant allele to protect it, not to express a trait. It will only take a homozygous recessive to express a trait. Okay. Does this make sense? Over there, table. Yeah? Explain dominant trait. Oh, and then you are laughing at it.
But
Okay.
dominant trait is the big letter, right?
It's
And then it's the one that's both.
It's the trait that is observable, that affects the.
With the osmosis?
No, wait, I'm thinking about something different.
Yeah.
Yeah.
Observable? Okay, okay. Extra credit points?
Okay.
Okay.
No, I'm gonna start getting stuff wrong.
This is the mom, we need a dad. Matthew?
Ah, dang. We're enemies right now. Just kidding.
Okay, good. So here you have mom and dad, okay? Matthew and what's his name?
You don't know my name?
I get confused. I met them, Matthew, they got married, they got happy, they met up in 1301 lecture. And they were like so happy with each other that during class they just like kept talking with each other, they made distractions.
You can skip that.
They were like so distracted that they didn't let other people like concentrate in class, boom. It happens, right?
So
And-
who are the kids?
Let's go for the kids. Let's go over there. Those
Come on, my
those four
children.
over here. Those four over here. Yeah, do you four over here. Over here.
Part white.
What's your name? Jackie, can you take out your jacket, please?
I promise I wasn't.
Over here, children,
I don't
over
know,
here.
two of these three.
Yes, over here, children.
Are you watching?
I
think so.
Over here.
On your paternity test,
Okay, right there. So let's bring you over here. Alright, so mom and dad, children, okay? Mom and dad, children, okay? Alright, so let's say that. Can I
I don't know.
Definitely it's
Okay.
paternity test.
Let's say that, just put it right here. Both of them are wearing black, right? They are affected, okay? They are affected. Parents are affected, they have the trait,
Why do you
okay?
use the hoodie instead of black? Like 3 of them.
Yeah, 3 hoodies and also here. He has a hoodie right here. Yeah. Right here, hoodie, right there.
Oh, yeah, that's true.
Okay? Okay. Don't question my affected, you're making me lose my thought. Affected parents, and then you have here, affected child, affected child, affected child. But one is not, okay? One is not. She's protected, okay? She's safe.
So that means we both have the recessive.
Yes, you are heterozygous, right? But the key point is that there is a dominant trait because you're heterozygous, you have the lowercase, because you will also have the uppercase, yes, the big one, and you are the same way. You give the big one to him, and to her. But you give the little one to her.
That doesn't mean we don't love you.
Oh, okay.
Hmm?
So they have to have both.
They are
Yeah,
heterozygous,
So
right?
that way she can come
So
out.
they're both
Yes.
big H, little h, big H.
That's
Because
right.
So
if
that's
we were
when
both, if there was only one little, I think it'd come in the second generation, right?
If it was like one homozygous dominant, then yeah, you will not be able to see this, right? But that's why you see this, right? Whenever you have a affected parent, and then you see an unaffected child, that's a clear giveaway. Heterozygous, one unaffected, hence it is a dominant trait. So, okay, hoodie, black, okay? Black, hoodie, right? The vast majority are affected, right? One unaffected. This tells you that it's a dominant trait. If the vast majority is affected, one unaffected, then it's clearly, right? It is a dominant trait, okay? Right, so whenever you see dominant parents, the parents being affected, the child, most of them being affected, but one is unaffected, boom, dominant trait, okay? All right, so now let's do the recessive trait, okay? So now let's say that this is the trait, but unaffected, okay? Unaffected. Like not showing the trait that is affected. It's unaffected, okay? Unaffected. So they are not showing the trait, okay? So
Why
now
don't you
they.
do glasses?
Hmm?
Why don't you do glasses?
Glasses, we have glass. We need one glasses for her. We need
I
two
was
glasses.
supposed to
Here you
have
go.
glasses for you.
Yeah.
I'll stop
Can't see. You can actually
Just
wear
in
glasses.
there.
I have my glasses, my bad.
Okay, we have
Like,
glasses.
oh, originally
We have
bossy.
glasses right there. Okay. Right, let's do glasses. You can put the hoodie away now. Yeah.
No.
Nice.